All right, thank you so much. It's really fun to be here, and it's just been a fabulous meeting. I'm going to introduce you to a neural organoid platform that we're building at STEM Farm, and one of the unique components of that is that we can incorporate microglia into the organoids, and that's why I'm here in the immune function. So the talk is going to be a little bit different than some of the other talks that you've heard recently. But neural inflammation is a really big problem in CNS drug discovery, and it's a matter of animal models just don't recapitulate, what's going on in vivo, as well as if you simplify the models too much, monocultures just can't tell you the whole story of what's going on. So the role of neural inflammation is also critical to many CNS indications, particularly things like neurodegeneration, traumatic brain injuries, ischemia conditions. So it's something that's really critical that we figure out. So advanced in vitro models need to incorporate neural cell diversity and immune components to adequately model neural inflammation. So that's what we're working towards doing at STEM Farm. So STEM Farm is an early stage company in Madison, Wisconsin. Our technology is coming out of Bill Murphy's lab at the University of Wisconsin. So he is a bioengineer and has developed synthetic biomaterials that really enable our platform. So we use PEG-based hydrogels, and I'll show you how we build these in a little bit, but PEG-based hydrogels that are really optimized to allow adhesion as well as ideal mechanical properties and the ability for the cells to remodel that substrate in order to generate these. So they're iPSC-derived, and so here, this image that we're showing, they're in a 96-well plate grown on top and within the hydrogel components. We start with neural precursor cells that then differentiate into multiple neural cell types, and I'll show you that. But then we add vascular and immune components. So the white cells in this case, so we have green that are astrocytes, red that are the neural cells, and then the white cells are the microglia. And this is IBA1 staining. The microglia tunnel into the organoids. They take on a very ramified morphology, and I'll show you a lot of data demonstrating that they're responding to effectors. So how we build these is in a 96-well plate. We start with our synthetic hydrogel plate, non-specified, high-quality neural precursor cells in a proprietary media, allow those to differentiate. Then we add the endothelial cells and supporting MSCs, and finally we add the microglia. So this model is being built as a developmental neurotoxicity model through funding through NIEHS, and that's the original manuscript from the Thompson and Murphy labs described that as well a few years back. So we can essentially build these and use them for developmental neurotoxicity within the context of 4 weeks. So from the NPC stage to adding treatments and doing the analysis. What's nice is that because they're in 96-well plate, they're ready kind of for screening applications. We can do dose response curves, and downstream analysis is available. So one of the important components is really to understand the cell types that are present in the organoid that we're building. And so we've been able to do single-cell RNA-seq. Again, these are single-cell data from day 21 organoids, really demonstrating the fact that we are generating a developmental neurotoxicity model. And you can see that we have, so the Tisney plots demonstrate that we have a lot of radioglial cells, we have intermediate progenitor cells, but we also have late and early neurons. We have a population of astrocytes, and then the cells that we add, the endothelial cells, the supporting MSCs, and the microglia. So we identified these cell clusters using the most differentially expressed genes and using enriched gene ontology terms to identify them, as well as to compare with in vitro cortical development. The organoids are very reproducible. So these are 3 different organoids that we then did single-cell RNA-seq on. Just grossly, you can see that the Tisney plots are really well represented. But then if we do Spearman rank correlation coefficients between the replicates, it's very, very tight. We've done a lot of bulk RNA sequencing as well, and we actually were part of the Illumina Accelerator a few years ago, and so that was really beneficial to us to be able to learn how to do these analyses and then do a lot of it while we were there. So we also did a lot of bulk RNA-seq analysis and find that we have very good replication between the organoids, and that's allowed us to really use differential expression analysis and gene ontology analysis to be able to look at perturbations with the organoids and make conclusions from that. So as I discussed earlier, one of the biggest differences of our organoids compared to some of the competitors is the ability to incorporate the microglia component. And here we've taken single-cell RNA-seq data and taken this cluster 7, which is the microglia component, and did transcriptional analysis compared to a transcriptional gene set that was identified in 2019, where they did a large-scale meta-analysis of primary microglia to determine really what are the gene signatures that should be in an in vivo situation. And of those 249 genes, we have 95% of those are represented in the context of the microglia in the brain, in our neural organoids. And that's one of the big differences. So if you take microglia out of the context, you take primary microglia out of the brain, they're going to behave very, very differently. Microglia in monoculture behave very differently, and they need these other cell interactions in order to function appropriately. So microglia biology is very complex. People like to talk M1, M2 phenotypes, inflammatory, neuroinflammatory, but the reality is the microglia, it's a kind of a kaleidoscope, really, of interactions that's going on and variable phenotypes. And the whole point of the microglia in your brain is to respond. And so they're ready to do that. And so the literature is pretty complex, I would say, about microglia. The fact that we can now have iPSC-derived microglia is really exciting, I think, to be able to look at different patient populations and then incorporate them into models such as this. So now I'm going to talk about a couple case studies that we've done around neuroinflammation and then neurotoxicity with our neural organoids. So in the brain, right, your microglia are in the resting state, very ramified morphology, ready to sense. When they have some kind of assault, like a bacterial infection, or a hypoxic event, they can become activated to an M1 kind of neuroinflammatory, and that's neurotoxic. On the other hand, treatment with things like IL-4, IL-13, or TGF-beta, IL-10 can induce a neuroprotective response. And then there's Plexicon has a compound 3397, which is a specific CSF1R inhibitor that has been shown to deplete microglia. That can be a good control compound for depletion of the microglia in the context of this. So we built the organoids, did treatment, and looked at responses. And essentially, from a transcriptional state, we can build transcriptional gene sets where we have an inflammatory signature, an anti-inflammatory signature, and then a perturbation of microglia signature. So the PLX3397 and actually valproic acid correlate very well with their gene signature in this case. And valproic acid, many know, is a HDAC inhibitor that's used as an anti-epileptic, but it does have some neurotoxic phenotypes, neurodevelopmental phenotypes, and in fact, people are using it to attenuate an M1 phenotype in clinical trials. So we can use our data set, both our bulk RNA-seq and as well as our single-cell RNA-seq analysis then, to look at specific genes that people are interested in. And this model is proving to be pretty interesting as far as target discovery in these kind of areas. So this is just pulling out some highly differentially expressed genes in a pro-inflammatory, anti-inflammatory, and then microglia depleted signature. So we can do immunoassays. So this is a Luminex multiplexed immunoassay demonstrating that we can, from the cell culture supernatant of these organoids, we can detect inflammatory cytokines, So a lot of responses, but it's not a general response because we have control compounds where we don't see a response. This is plus or minus interferon gamma and LPS stimulation. And then what we've been able to do is look, in this case we used GFP-tagged microglia in the context of the organoid and asked if we can look at morphological metrics in the context of the organoid. So this is using confocal microscopy, and here looking at a sphericity measurement. So in the case of a ramified microglia, they don't look like a sphere, right? They have a very low sphericity measurement. You can see we're spinning this, and you can see the ramified microglia. As they move to a more active amoeboid shape, they become more sphere-like, and we can measure that. So in the context of the organoids, so this is confocal microscopy looking at an organoid. So blue in this case is the nuclei, purple is NCAM1, so looking at the neurons, and then the green is the GFP-expressing microglia. So a couple points here. One is that the microglia are really nicely distributed throughout the organoid. And if you look at brain sections, that actually happens too. The microglia really space themselves in a manner that's controlled. They have a nice ramified projections. They're tunneling in, and importantly, if you look at the XZ or YZ panels here, we're definitely in the context of the organoid. So our organoids are built on top of the hydrogel. They're pretty big. They're like 1 millimeter wide, but they're about 150 microns thick. And so that makes some of this imaging analysis helpful, and it also reduces a necrotic core. So we don't see a necrotic core in these organoids. So these are the control. And if we treat them with interferon, the microglia respond very differently, take on an activated larger congregating morphology. You can see that they're actually moving into the hydrogel. So this is the hydrogel here. In this case, the purple is GFAP, so it's astrocytes. Both astrocytes and the microglia in this case are activated. And if we look at LPS plus interferon, they have a different morphology, but still we see areas where the microglia are activating, so we're doing analysis, morphological metric analysis, as well as time analysis on this. And next year I hope to have a lot more data around that to show you. So now we have a case study around developmental neurotoxicity. And in this case we took 4 known neurotoxins, chlorpyrifos, lead acetate, valproic acid, and then that PLX3397. Did dose response curves on NPCs to choose dosing, and then dosed over the course of 4 days with these neurotoxins, and then isolated RNA and did transcriptional analysis. And what we see is we get predicted responses. So chlorpyrifos is an organophosphate pesticide, which has a well-described mitochondrial phenotype. And here we see an upregulation of gene ontology sets affecting microglia. We see a hypoxic response as well as an angiogenic response. So remember, the endothelial cells are there. So endothelial cells, we can pick them up in the neurotoxic screen. We also see a large decrease in neurogenesis and metabolism. With lead acetate, we see a downregulation of ECM and cell motility components. With the valproic acid, as I described, we see a decrease in the neuroinflammation response, which is correlated to the PLX3397. So we're funded by NIHS to build this out further. We're working on some different transcriptional analysis to kind of streamline things so we don't need to do full RNA-seq on all of the data sets. So we feel that we're at a convergence where we have unprecedented access to relevant human neural subtypes. We have these biomaterials that can mimic the physiological environment, and then multimodal measurements and the ability to interpret these large data sets. So we're looking to partner with companies to look at CNS diseases, particularly around target microglia and neural inflammation pathways, target discovery, phenotypic screens, neurotoxicity, and then our expertise, again, is in biomaterials. And so I have a poster this afternoon, 2 posters are right next to each other. If you have any biomaterial needs, be happy to talk to you about that. And just some, our team doing a great job in Madison, Wisconsin. We had wonderful collaborators at the Illumina Accelerator that helped us really with a lot of the correlation, getting the technology up and going. And then we have access to the UW confocal imaging cores, which is very helpful. Our funding's been from NIH, NIEHS, and NINDS. So thank you very much.